All right, well, thank you for that introduction, and good morning, everyone, and welcome to the 2019 SBIA Generic Drug Forum. I hope everyone had a great time yesterday and is well rested for another day of fun today. And my name is Varun Vasudeva, as they mentioned, and my co-presenter here today is Aiden Nguyen, and we are both primary filing reviewers within the Division of Filing Review. And our presentation here today will cover filing and refuse to receive. So today's topics of discussion are deficiency metrics, and we want to share with you some major and minor deficiencies that within each module from the filing perspective. And finally, we would like to conclude our presentation here today by inviting all of you on a journey to the great beyond. Some refer to it as an enchanted forest. Others refer to it as a magical place where AMDAs are accepted for filing. Some of you may have your own name for it, but whatever you want to call it, we would like to end our presentation here today with a case study and dive a little deeper into the life of what a filing primary reviewer looks at. And to start off, we want to show you a chart of some RTR statistics over the past few years. As you can see, the number of applications that have been refused has decreased significantly, as alongside the total number of AMDAs that have been received have increased. Some of the reasons for the decrease in numbers are attributed to an update to the guidance in December of 2016, posting the AMDA filing checklist map, and finalization of the content and format guidance. So our take back from this is industry as a whole is learning from events such as this SBIA. And for your reference, we have provided some of the top reasons that AMDAs were refused in fiscal year 2018. These include dissolution, when an application has 10 or more minor deficiencies, impurities, stability data, Q1, Q2, and other categories. Now to scratch the surface on some of the other categories, they are as follows: incomplete Type 2 DMF completeness assessment, inadequate packaging, incomplete and/or failed BE studies, and lack of master batch records. I would now like to highlight some of the deficiencies found within Module 1. Minor deficiencies that would result in an information request include incomplete form FDA 356H, basis of submission, and labeling. Major deficiencies that would result in an RTR are an unsigned or an omitted FDA form 356H. Now let's dive a little deeper into form FDA 356H. Some of the common deficiencies we see here are found in Field 9. If the drug product is subject to USP, revise Field 9 to include USP. Field 11, list the full chemical drug name. If it does not fit in the space provided, we typically see this in the continuation page. Field 20, most mistakes are found in this field. The applicant will check the wrong box on the form and claim another patent certification. When this happens, they typically have to revise Field 20 or the patent certification. Sometimes we see that the applicant checks off the wrong box accidentally in their information request response. They should also include patent certification in the title of the statement in Module 1352. Field 28, the information is inconsistent. An example of this would be an incorrect phone number provided for the contact person. And Field 29, a typo of the DMF, or they failed to list the DMF that's found in Module 142. And at this time, I would like to give a kind reminder here to use the most up-to-date forms that are at the time of submission that are found on the forms website. Module 112.11, basis for submission. Here you should use the designated RLD or RS that is currently listed in the Orange Book. Is a suitability petition required? If yes, the applicant should provide the docket number as well as a copy of the FDA's correspondence approving this petition. Also regarding suitability petitions, if an ANDA is submitted that is proposing a change from the RLD that is permissible under 505 J2C, but for which the suitability petition has yet to be approved, then DFR will RTR the ANDA, meaning this could be considered a major deficiency. And this information can be found on regulations.gov. Module 114, labeling. Legibility issues for container closures could be avoided by providing photographs with higher resolution of the container closure. And you may want to consider providing larger photographs and limiting them to 1 per page. The recommendation here is to provide a proposed container and carton for each strength and the packaging configuration. Moving on to deficiencies found within Module 2. Minor deficiencies that would result in an IR are to provide, the applicant should provide PDF and Word files. In Module 2.3, we're looking for each drug substance and each drug product. And in Module 2.7, for the summary data tables. Major deficiencies that would result in an RTR are typically found in Module 2.7 under the dissolution table 5. Module 2.7 deficiencies, summary data tables. An applicant should provide the COA for each strength and the RLD. And for Table 10, it is recommended to provide the exact location of the LTSS study reports, including the module, section, subsection, pages, as well as working hyperlinks. Dissolution studies in Table 5. This is a common area for major deficiencies where DFR will RTR an ANDA. Dissolution not conducted on 12 units. Dissolution on all strengths not in the original submission. Dissolution not conducted in all test media per the draft guidance or OGD recommended medium. And dissolution not conducted in all test media per the scoring half-tablet studies for modified release dosage form not in the original submission. And that wraps up module number 2. I'll now pass it on to Aiden. Thank you.
Thank you, Varun. Good morning, everyone. I'll now be talking about the deficiencies that we'll find in module number 3. For those of you that don't know, module number 3 is a hotbed for a lot of the minors as well as the major deficiencies that we will see in an AMDA. So I know it's early, but stick with me and I'll try to make this as painless as possible. First, we're going to touch upon some of the minor deficiencies, which will be formatting of justification tables, as well as the regional batch information. Then we're going to dive deeper into the major deficiencies, which will be Q1, Q2, justification of impurities, and stability. Inside module 32S45 and 32P56, the applicant should provide a justification table for the impurities. 32S is for the drug substance, while 32P is for the drug product. It is recommended that the applicant follows the templated format to ensure that the reviewer have all the required data that they need. It's also recommended that the applicant provide separate tables for the 3 types of impurities, which are specified identified, specified unidentified, and unspecified impurities. Inside module 32R, we will find the batch record as well as the reconciliation tables that should include theoretical, actual, and packaged yield. Commonly, we can see a minor in which case the package yield is not expressed in the correct product units. For example, for oral dosage form, the package yield can be expressed in the number of bottles. However, the product units is actually the number of tablets or capsules. So there should also be detail about how many tablets or capsules was packaged per batch. Per 314.9489, the formulation should be qualitatively and quantitatively similar to the RLD. What we mean when we say this is that your formulation should use the same inactive ingredients or other ingredients as the RLD. There are some excipients where changes are permitted, such as buffers, preservatives, or antioxidants in the case of parental dosage form. We highly recommend that before your submission of your ANDA, you submit in a control correspondence inquiring about Q1, Q2. Now, the second Q in Q1, Q2 stands for quantitatively the same as the RLD. This is essentially your plus and minus 5% range in the amount you use versus the RLD. It is important to note that from the range of 100 to 105%, you are considered quantitatively the same. However, we also suggest that you take it one step further and justify that amount using the IIG. This is because even though you are quantitatively the same, you still run the risk of a potential RTR if you use an amount that exceeds the upper limit for that ingredient. Revisiting the justification of impurities table, we will now specifically talk about how we justify for those impurities. For specified identified and specified unidentified impurities, there should be a justification for any impurity where the acceptance criteria exceeds the regulatory threshold. You can justify these impurities using scientific literature, toxicity studies, if that impurity is a major metabolite of the drug substance or staining levels seen in the RLD. For unspecified impurities, the acceptance criteria should not exceed the regulatory threshold. Now we have to talk about the monster in the room, which is stability. We are still seeing non-adherence to the recommendation of 2 API lots per strength. This was communicated in the stability guidance as well as the stability Q&A. It is important to note that your ANDA does not need to have 2 API lots per batch, but it is 2 API lots per strength. There should also be a minimum of 3 test batches per each strength. Overall, most of the ANDAs that we receive, we do see the intention for 3 batches per strength. However, sometimes data can be duplicated, where data for one batch can be submitted twice, resulting in us missing data for the 3rd batch. This can result in a potential RTR due to the failure of meeting the recommendation of minimum of 3 test batches per strength. In the Q1A R2 guidance, it is stated that if an applicant noticed significant change under the accelerated condition, intermediate studies should be generated and 6 months worth of data be submitted with the ANDA. It does not state, however, that you should stop your accelerated study. So what we actually recommend is that you continue with the accelerated study up into that 6-month mark and then submit in 6 months of intermediate, 6 months of accelerated, and 6 months of long-term data. For liquid as well as semi-solid dosage form, we also recommend you submit in 6 months worth of worst case as well as non-worst case orientation studies. And please verify all start and pull dates for those stability studies to ensure that it does fulfill the 180 days worth of data recommendation. Okay, after you make it through all the pitfalls in module number 3, now you make it to module number 5, you're in the home stretch. We don't see many major deficiencies in module number 5, and some of the minor deficiencies that we're going to lightly talk about today are data tabulation data sets and a couple ECTD issues. Inside module 5312, we are looking for the pharmacokinetics data to show bioequivalence for your drug product. Specifically, we are looking for pharmacokinetics and concentration data sets. These data sets should be provided in XBT format, and commonly we can see duplication in this area, where pharmacokinetics data is provided twice, and we're missing the concentration data sets, or data from the fasting study is provided twice, and now we're missing the data from the Fed study. So please check for duplication to ensure that all the recommended data is provided with your AMDA. Module 5 contains a lot of big files. Some of these folders can contain 10 plus documents, and each document can contain hundreds of pages. So it is imperative that we follow ECTD guidance to ensure a faster review. Descriptive leaf titles are extremely important. There should be a bookmark for any documents that's 5 or more pages, and those bookmarks should also match the table of contents. Okay, so we made it through all the modules. We have highlighted some of the minors as well as the major deficiencies that you will see in each module. Now we would like to invite you guys on a journey, kind of like a day in the life of the glamorous DFR reviewers. We will touch upon some of the things that Varun and I have talked about to give you guys a perspective of what we see while we are reviewing the AMDA. So without further delay, let's jump into it. This is the view that we see when we first open up your AMDA. It is broken down into modules in the top left, and you can view selected documents in the preview screen. So let's jump into module number 1 first. Inside module 1.12.11, the basis of submission. This is where we get the background information to see what your AMDA is based upon. For this example, the reference listed drug is known as the Super Soldier Serum. So some of you are smiling because this should sound very familiar to all the Marvel fans in the room, because to me, this is always Captain America's steroids. So this is how Captain America grew from 5'2" to 6'2" within 30 seconds of screen time, right? So the dosage form is an injection, so that matches the movie, so it's verified, that's good. The applicant is the Avengers. They're one of our regulars, so we have meetings with them all the time. So that's a regular. It looks like all the information is provided. However, let's look more closely at the suitability petition section. This AMDA did require a suitability petition, so a docket number as well as a letter of approval should be submitted with the AMDA. And if it is not, as in this example, it will be an IR. Now let's jump into module number 2. Inside module 2.7, we are looking at the clinical summary in the top left. Specifically, we are looking at the summary tables. We note that the summary table is provided in PDF format, which is good. However, it's also recommended that the summary tables be provided in Word format. And if it is not, then it will be another IR. Now we are looking at summary table number 10 inside module 2.7. This is where we get the background information to the BE study that was conducted for your AMDA. Now from the study title, one of the sentences says, transform a Steve Rogers into a fighting machine with no sense of fear. I am very interested in this drug and hope that it makes it to the public. However, it also shows that the study population is only 1 person, so it's not very reliable, so I wouldn't expect this to come out anytime soon. The clinical site, it says Black Site X. That makes me very disappointed because I was really looking forward to also getting the address and phone number to this site. However, the Avengers like to keep things classified, so we let that slide for them. Now let's look more closely at the LTSS data on the bottom. Inside table 10, it is recommended that you have a working hyperlink or at least point out the exact location for the LTSS data. If it is just a general statement like module 5, as in this example, it will be another IR. Now let's jump into module number 3. We are inside module 32P56. We are reviewing the unspecified impurities table. We note that the table does not appear to follow the template, and comparing it to the template, it looks like the IT category is missing. So this will result in another IR. Upon further review, we also noticed that a USB monograph was used as a justification for unspecified impurity. This can result in a potential RTR according to the impurities guidance. While reviewing the stability protocol, we noticed a troubling sign. It appears that the accelerated condition was not conducted at the 6-month mark. Digging through the AMDA, we found the justification. And to summarize, it looks like the applicant noticed a significant change at the 3-month mark of the accelerated condition. So they submitted in 3 months of accelerated, 9 months of intermediate, and 9 months of long-term data. This can result in a potential RTR due to the failure of not meeting the recommendation of having at least 6 months worth of accelerated condition stability studies. Now let's jump into the last one, module number 5. So inside module number 5, we're very close to the end, and we open up file number 1, and we see this. So unfortunately, page number for bookmarks are not very effective or descriptive. So this will result in an IR. Hoping for better results, we open up file number 2. And again, the applicant, the vendors have failed us